Good afternoon, everyone. I would like to welcome you to the National Fragile X Foundation's webinar, Genetics 101, Navigating the Many Twists and Turns of Fragile X Inheritance. I'm Jane Dixon-Weber, the Director of Education and Support Services at the Foundation. And these webinars are made possible because of your contributions to the Foundation. A couple housekeeping issues. Brenda Finucane does have a presentation she's going to give that will take about 40 minutes, and then she will take questions. Use the chat box on the side for your questions, and also use that chat box if you have any problems. Now, I would like to introduce today's speaker, Brenda Finucane. Brenda is a licensed genetic counselor and the Associate Director of the Autism and Developmental Medicine Institute of Geisinger Health System in Lewisburg, Pennsylvania. Ms. Finucane has a long-standing clinical and research interest in Fragile X and other genetic disorders associated with complex intellectual and neuropsychological symptoms. She serves on the Foundation Board of Directors and on the Scientific and Clinical Advisory Committee of the Foundation. She's also a consultant genetic counselor for the Foundation. She's the lead author on the National Society of Genetic Counselors Practice Guidelines for Fragile X Testing and Genetic Counseling. Good afternoon, Ms. Finucane.
Thanks, Jane. So nice to be invited to talk about genetics when talking with people and families who are living with Fragile X syndrome and Fragile X associated disorders. I'll start by saying that even for families who've been through genetic counseling, very often it helps to have a refresher about the genetics of Fragile X because this is one of the most complex genetic disorders in terms of explaining its inheritance. As Jane mentioned, I will just give a few disclosures. I am a consultant and I'm on the board of the directors of the National Fragile X Foundation. I'm also a consultant for LabCorp, and I'm also work as part of my day job for Geisinger Health System's Autism and Developmental Medicine Institute, including our Fragile X Center there. So I'm going to talk today about not only Fragile X associated disorders, or FXAD, but also the gene FMR1 and its various twists and turns and expansions that go on to give these different disorders. It's a very complex inheritance pattern, as was mentioned before, in that there are differences in how the gene can express itself depending on the size of the expansion, how many CGG repeats somebody might inherit, and also whether you're a male or a female. It's an X-linked disorder, as I'll explain, because it involves the X chromosome, and there are differences between males and females with regard to the X chromosome. The FMR1 gene can expand if there is a premutation to a full mutation, and there are also even smaller expansions known as intermediates that can eventually become premutations. So the bottom line is that the gene expansions that result in Fragile X associated disorders happen over multiple generations. So unlike many other types of genetic disorders that can just occur out of the blue in a family, Fragile X really doesn't occur out of the blue. It's very possible in some families not to see previous people with Fragile X syndrome or other disorders, but it doesn't mean that the gene expansions aren't happening. It just could mean that people are carrying expansions and not even realize it. In order for Fragile X to result in a large enough full mutation, a very large expansion that causes Fragile X syndrome, it has had to go through multiple generations of getting bigger and bigger over time. So that's why we say it's a multi-generational disorder, and when I, as a genetic counselor, first meet or diagnose a person with a Fragile X associated disorder, I know right then and there that it's not just that one person that I need to be thinking about, it's the entire family. So when we talk about Fragile X associated disorders, or FXAD, we're talking primarily about three particular conditions: Fragile X syndrome, FAXTAS, Fragile X associated tremor ataxia syndrome, and FAXPOI, Fragile X associated primary ovarian insufficiency. These are three very distinct disorders, medical conditions, that are due to different types of expansions in the FMR1 gene. So just for review, Fragile X syndrome is the most common inherited reason for intellectual disability and autism. It's found in all populations, all ethnic groups, and it occurs in families who don't have a history of developmental disabilities, as well as in families where there's a very strong history of developmental disabilities. As mentioned before, even in families where there's no previous history of Fragile X syndrome or developmental problems, the gene is still in those families in an expanded form because it takes multiple generations to get to the point of a full mutation. X-linked inheritance just means that because the gene is on the X chromosome, there's going to be a difference in the way it expresses itself in males and females. And in the case of Fragile X syndrome, both males and females can be affected by developmental disorders, but it's more common to have this happen in males. And of course, there's a very wide range and varying severity of the developmental and behavioral effects of Fragile X syndrome. So even in a family where you have two brothers who have Fragile X syndrome and both have Fragile X full mutations, you can have very, very different presentations in those two young men. Apart from Fragile X syndrome, which is the most well-known of the Fragile X associated disorders, we also know that Fragile X primary ovarian insufficiency occurs in women and girls, and this is a condition that's related to having a Fragile X premutation, which I'll explain shortly. But the features of FAXPOI include low ovarian reserve, which means that there are insufficient numbers of eggs, or not the usual number of eggs in the ovaries that one would expect in a female. And, you know, in the general population, as women get older and past menopause or near menopause, they don't have as many eggs as they used to. What we know in FAXPOI and is that this low ovarian reserve can happen in young women, relatively young women, even down in the early 20s, that they don't have a sufficient number of eggs, and that might lead to infertility. They may have problems with their periods, and they may go into menopause quite early. And about 25% of women who carry a Fragile X premutation are affected in some degree by FAXPOI. FAXTAS, which is Fragile X-associated tremor ataxia syndrome, is a neurodegenerative condition, meaning a condition that affects the brain, and it's in about half of males and about 15% of females who have premutations in the Fragile X gene. Now, what's really important here, because I hear many families worrying about this, is that this is not a condition, FAXTAS, that affects individuals who have Fragile X syndrome or full mutations. This is only a condition that we see in individuals who have premutations, so it's a carrier condition. It's progressive. In other words, the symptoms can slowly get worse over time, and the average onset is in the late 50s typically. And so you may have a grandfather of a child with Fragile X who starts in later life to have some shakiness, otherwise known as tremors, balance problems or ataxia. They may have some personality changes and psychiatric symptoms. And then with FAXTAS, there is a very specific MRI finding, but it's not found in everybody who has FAXTAS, and so the absence of this particular finding does not rule out FAXTAS. So again, these are the Fragile X associated disorders. But there are also plenty of other symptoms that can be found that don't fit neatly into these other disorders. So for example, there are some females who have Fragile X premutations, who are carriers of a premutation, who have social anxiety or shyness. Sometimes they have some problems with their mood, and so they may be very elated and happy one day, and then, you know, they may have depression at other times. They don't necessarily fit into, and they don't have Fragile X syndrome, they don't necessarily fit into one of those disorders, but they're having some symptoms from the premutation. There's also been some reports that some people with premutations may have an increased chance for immune disorders like thyroid disease and lupus. These are common disorders in the general population, in women particularly, and having a Fragile X premutation may increase this chance somewhat over the general population chance. We also know that there have been some reports that kids who have premutations or intermediate alleles, which are even smaller repeat sizes than premutations, Most of them develop very typically, and you don't see any issues with them, but every once in a while we'll get case reports of an individual, a male or a female, who's got a pre-mutation, who has ADHD or maybe autism or maybe an intellectual disability. And the verdict is really still out as to whether this is just coincidental or whether it's really related to the pre-mutation. The reason that many feel that this is very likely to be coincidental is that autism, for example, and ADHD, these are very common conditions generally in the population. And because having a Fragile X premutation or carrying that is very common too, the chance that you may have a child who just coincidentally happens to have ADHD and also has a premutation is not that far-fetched. On the other hand, we also want to make sure we're not overlooking something. And so right now it's not really clear, but we think that there may be some symptoms in children and adults related to developmental disorders like ADHD and autism, etc. However, the chance of this happening is at most only slightly increased over the general chance that that would happen in the population. So now down to the nuts and bolts of the genetics. How is it that a certain gene, FMR1, is associated with all these different kinds of things going on in families? Well, to go back to high school and college biology and to just orient everybody to what we're talking about, genes are chemicals really in every cell that are made up of DNA, or deoxyribonucleic acid, which is a body chemical. And FMR1 is a gene, and we have in our bodies and in our cells over 20,000 different genes that determine many things about us: our eye color, our blood type, height to a great extent, medical conditions that we may be vulnerable to a certain extent, our behaviors and our personalities. So there's a lot that are controlled for by genes, and genes are there at the moment a child is conceived. When a dad's sperm and a mom's egg get together, those egg and sperm getting together form the first cell that ultimately becomes the blueprint for the body. The genes really are the blueprint for how that body is going to form, and then genes also express themselves throughout life after a body is formed and a baby is born, and can continue to control body functions throughout life. Genes come in packages in the cell that we call chromosomes, and even though we don't use this technology very often anymore, it's useful to be able to picture chromosomes, because I want to point out to you really what we're talking about when we refer to Fragile X as an X-linked disorder. So in the cells, if you were to look at a person's chromosomes, which essentially are packages of genes, they would look like this, and you can actually visualize them using a microscope. And as you'll see on this picture, they're kind of scattered in the nucleus of the cell, and they are structures that have stripes on them that we refer to as bands. So if I was to take that picture and line up all those chromosomes very neatly, what you would see is that in this picture that we have a female, and she's got 46 chromosomes, or 23 pairs. The 23rd pair here, being pointed to by the arrow, are the sex chromosomes, and they determine if somebody's going to be a boy or a girl. In this case, there are 2 X chromosomes because she's a female. In this next slide, this is a male. He's got only 1 X chromosome and another chromosome called a Y, which contains genes for maleness and male traits specifically. So right off the bat, we know that because the gene for Fragile X disorders, FMR1, is located on an X chromosome, there are going to be differences between males and females. Females, in a sense, are protected from many of the effects of FMR1 expansions because they have a second X chromosome. Males only have that 1 X chromosome, so if they're going to have a problem with one of their X's, they don't have a second one to back it up. So Fragile X syndrome is considered an X-linked trinucleotide repeat disorder. X-linked refers to the fact that the gene FMR1 is located on an X chromosome. A trinucleotide is a series of 3 body chemicals together, and so cytosine, guanine, guanine, or CGG, is a trinucleotide, and these trinucleotides repeat in Fragile X over and over again. So you can have a series of CGGs in a row, and that's how this gene is composed. There are other sections of the gene, but a certain area of the gene has this CGG repeat pattern. So part of the gene includes this repetitive section of CGGs, and in most people, the CGG repeat number is 29. So they have 29 of these CGGs in a row. It's variable, and it can be anywhere from much lower than 29 up to somewhat higher than that. So if you have Fragile X DNA testing, they can actually check the number of CGG repeats that a person has. And most people have anywhere up to just below 45 repeats. So under 45 repeats in this FMR1 gene is considered a negative or normal result, with the average being around 29. I'll skip over to full mutation, and a full mutation is what happens when you have a very large number of repeats, over 200. And if you get over 200 of these CGG repeats, what happens is the gene doesn't function like it's supposed to. It doesn't produce its normal protein, and you have Fragile X syndrome. And so most people who enter our Fragile X community do so because they have a child or a relative who has been diagnosed with Fragile X syndrome and has been shown to have a full mutation with more than 200 repeats. A premutation happens when there's not over 200 repeats. However, it's much larger than we would expect in the population. And premutations have anywhere from 55 to 200 repeats. And then even below that, there is a category that we call intermediates. And intermediates are not a truly negative result or normal result because they're somewhat bigger than usual, but they're really not Fragile X disorders either, because they're not associated, as far as we know, with any kind of clinical symptoms, and also there's not a chance that they're going to jump up and expand in the next generation to a full mutation. So more about intermediates in a little while, but these are the categories of Fragile X expansions. So what does it mean if you have these different numbers? Well, as I mentioned, if you've got a full mutation, that means you have over 200 repeats, and we know that that's associated with Fragile X syndrome or developmental concerns. So, for example, the vast majority of males, because they only have one X chromosome, if they have a full mutation on their only X, they will end up having Fragile X syndrome. And that in itself can have some variability, but there are some developmental issues one is going to expect with that. In females, about half of the females who get a Fragile X full mutation, over 200 repeats, are going to have intellectual disability and Fragile X syndrome. But then the other half, many of those females, those girls and women as well, have some learning disabilities, have some other issues and developmental concerns, even if it doesn't fall into the category of an intellectual disability. There's a small number of women and girls who have full mutations who seem to be almost perfectly compensated for by their second non-Fragile X, and who really don't have a strong history of learning problems, have not needed supports in school. But in females who have full mutations, that's actually the minority. Most would have some evidence of either learning disabilities or full Fragile X syndrome. If somebody's got a premutation, so that is an FMR1 gene that's expanded from 55 to 200 repeats in that range, then they are at risk for having FAXTAS and FAXPOI, those other types of Fragile X associated disorders. And for them, there are a number of considerations. One is that they may develop symptoms of FAXTAS, or if it's a woman, she may have problems with infertility or FAXPOI. And when they go on to have children, there are reproductive considerations. Is that gene going to expand to a full mutation in the next generation? Are they going to pass that on to their children, etc.? We also know, and again, the literature is a little mixed on this, but there may be an increased chance for some developmental, behavioral, and psychiatric concerns, even in individuals with premutations. At this point, given the general population chance of having a child with autism or ADHD or intellectual disability out there, this premutation may only slightly increase the chance that that might happen over the general population risk. And then finally, this intermediate category where you've got more than expected, a higher than expected number of CGG repeats, but it doesn't come up to the premutation range. There's really no risk for Fragile X-associated disorders with intermediates. In these families where we find these intermediate Fragile X or FMR1 expansions, the gene may be very stable and it may just be a family trait, and it's like that for years and it never expands into the range of Fragile X-associated disorders. However, in some of these families, it's important to watch because it could be that you have a woman, for example, who has an intermediate FMR1 expansion of 48, and then she goes on to have a child, and her child then has a premutation, and then that child goes on to have children in the future and may have a child with a full mutation. So over multiple generations, it may eventually become a premutation. And of course, under 45, so 44 and below, they are not at risk for Fragile X-associated disorders. So another way of looking at this on this slide here, you'll see that I've just talked about the normal range below 45. I'm not going to mention the intermediates here, but full premutations and full mutations, there's a different way we can look at this, and this is in terms of what's the mechanism? What do we know about how these different expansions of the FMR1 gene can cause somebody to have Fragile X-associated disorders? So let's say this is the gene here with FMR1, and in the normal range, typically what happens is you have the FMR1 gene, which produces Fragile X protein, FMRP, and in order to do that, it produces an intermediate type of body chemical called messenger RNA, and messenger RNA is really important in trying to translate the gene code into this protein. And this is the typical way that that is supposed to happen. And what happens in Fragile X full mutations, to jump over here, is that this gene has over 200 CGG repeats, and when that happens, the gene becomes what's called methylated. There's a chemical tag that gets attached to it that kind of shuts it down, and it doesn't really produce protein. So you're missing Fragile X protein. It's really not producing any substantial amounts of messenger RNA, and so you're really not having any protein produced, and that's when you see Fragile X syndrome. Fragile X syndrome is a disorder caused by the lack of Fragile X protein, or FMRP. What's interesting in the premutation range is that people who have that 55 to under 200 range of CGG repeats, they do produce protein. Maybe not at exactly the same levels that somebody who doesn't have a Fragile X expansion, but they're certainly producing plenty of protein for what they need to do in their daily lives and their brain functioning. What happens with the premutation, however, is that because this gene is larger than it should be, because it's expanded, when it goes to produce protein, it has to churn out lots of messenger RNA in order to make that protein production happen. And when that happens and they're producing lots of messenger RNA, what we see happening is a person may end up with FAXPOI or FAXTAS or some of these psychiatric, behavioral, and developmental symptoms that I talked about before. So the bottom line is there's two different disorders going on in Fragile X, and one has to do with the lack of Fragile X protein, and that's what we see in Fragile X syndrome, but the Fragile X associated disorders of FAXPOI and FAXTAS, etc., are not due to lack of protein. As far as we can tell, we think the mechanism has more to do with this buildup of messenger RNA, and so something that gets referred to as messenger RNA toxicity. The cells are being made this is toxic to the cells, or causing cell death to have too much messenger RNA. So for these Fragile X expansions, and we like to use the word expansions now rather than mutations. We're still calling them full mutations and pre-mutations, but really the genetics world in general is moving away from this word mutation, which in many ways has a negative connotation. So we'll call them FMR1 expansions. And what happens with Fragile X expansions is there's two different genetic problems going on. We've got Fragile X syndrome due to a full mutation and those full mutation symptoms. And with that, Fragile X full mutations become methylated, and then they don't produce their protein. FMRP stands for Fragile X mental retardation protein. It was named many, many years ago, and that terminology was in place at that time. So FMRP is not produced, and Fragile X syndrome is caused by the lack of FMRP. FMRP is really an important protein for brain functioning throughout life. Now, one really important thing to know about Fragile X syndrome and full mutations is that the severity of Fragile X syndrome is not correlated with the size of the full mutation. That's a question that families understandably ask all the time when they get a new diagnosis. They may say, well, what does it mean that my child has 300 CGG repeats? And does that mean he's going to do better than a child with 1,000 CGG repeats? And the answer is no. Once the repeat number exceeds 200, the gene is shut down, it doesn't make its protein, and it's an all or none phenomenon. So in other words, even if you have somebody with 1,000 CGG repeats, that gene is shut down, and somebody with 250 CGG repeats, that gene is shut down just as much as the one with 1,000 repeats. So it's an all or none phenomenon. The number of repeats over 200 really does not predict what the symptoms will be or the severity of Fragile X syndrome. So the other genetic problem that I mentioned has got to do with the associated disorders of FAXPOI, FAXTAS, and then those premutation symptoms that we see sometimes go along with people who have premutations. We know that FMR1 premutations can produce normal amounts of FMRP, so the problem is not the lack of the protein, but the large number of CGGs cause an increase in that messenger RNA as the gene is trying to make the protein. And Fragile X associated disorders are therefore thought to be due in large part to FMR1 messenger RNA toxicity. The reason this is really important, well, there's many reasons, but a key reason why we want to make sure people understand this is because there's a lot of work going on now on therapeutics and drugs for Fragile X syndrome. Drugs that might eventually be able to replace the function of that FMRP, that protein that's missing. And it's important to realize that with FAXPOI and FAXTAS and the premutation disorders, that strategy is not necessarily going to work for those treatments. They need a whole different kind of angle because the issue for premutation disorders has to do with messenger RNA toxicity, and so it would really be a different approach to treatment. So as if this wasn't complicated enough, and my heart goes out to all those families out there dealing with Fragile X because it is very complicated. You have to learn a new vocabulary, you have to understand these different mechanisms, but then again, we also know that there's differences in males and females on how Fragile X gets passed down in a family. As a genetic counselor, I work with lots of different genetic conditions, including many deletions of chromosomal material or extra chromosomal material, and much of the time, all I need to say to a family with one of those other disorders is, well, there's a missing piece of genetic material, and then we're kind of done the genetics discussion. That's never the case in Fragile X. And a lot of times, families and couples are learning about Fragile X for the first time when they are hearing that their son or daughter has a disability related to Fragile X. And so this is an amazing amount of information to have to take in all at once, and it's not at all surprising that many families have no recollection about what they learned about Fragile X inheritance. And it's only after some time that they're able to take a breath and say, okay, I really didn't catch that. How do I learn about this again? I will say, as a genetic counselor, it's completely acceptable to return and see a genetic counselor later on to have a discussion about inheritance if you didn't catch it the first time, as most families and couples do not get that information the first time and they do not absorb it at the time. So I would encourage you not to feel any way ashamed or that you didn't understand what was going on. This is an extremely complicated genetic disorder, even for genetic counselors. I have genetic counselors who contact me regularly to just check on their understanding of Fragile X inheritance. It is one of the most complex disorders to explain. So with that being said, what do we know about how these expansions are passed down in families? Well, we know that males and females can pass FMR1 expansions to their children. However, women, when they go on to have children, will pass their X chromosomes, and they have two of them, so it'll either be their Fragile X chromosome or their one without the Fragile X, and they can pass it on to a son or a daughter. Men, on the other hand, they only have one X chromosome, and when they go on to have children, they're either going to pass a Y chromosome, which will go to their sons, or they'll pass an X chromosome, which will go to their daughters. So men who have Fragile X expansions will pass their X to all of their daughters, but none of their sons. And there are a few things we can say absolutely in Fragile X, and this is one of them, that there's no male-to-male transmission. So as a genetic counselor, when I sketch out somebody's family tree and I see that, you know, a mom has a brother and he has a son, and their family's wondering about who should be tested for Fragile X, at least I can say, well, in terms of this Fragile X gene in the family, there's no need to test that son because it could not have gotten passed down from a father to a son. So that's one of the few things we can say for sure. What do we know about what happens to the CGG repeat series when it's passed down? So this expansion, well, it depends on the gender of the carrier. So if a woman has a premutation, it can expand to a full mutation in her sons or her daughters. Or when she passes it down, it could stay the same size or just expand a little bit and stay in the premutation range. On the other hand, if it's a male who has a premutation, when he passes it down, usually it's going to remain pretty stable, and we don't see large expansions typically when a father passes his Fragile X expansion or his premutation to his daughters. The other variable is the number of repeats. The larger size repeats in the premutation range in females have a much greater risk than the smaller ones of expanding to a full mutation. So for example, let's say you're a woman and you have 140 CGG repeats, you have a Fragile X premutation. The chance, if you become pregnant, that if that's passed down, it will expand is 100% for that particular Fragile X gene because it's already pretty high up in the premutation range. And once they get over around 100, in the next generation, those Fragile X premutations that are large are going to expand to a full mutation. On the other hand, if you're a woman and you have a repeat that's down in the smaller range, 58 repeats for example, there's an overwhelmingly high chance that that's not going to expand all the way up to a full mutation. It'll probably expand, but it may expand further into the premutation range, but not yet get over 200 repeats. So the larger size repeats in females have a greater risk than smaller ones to expand to a full mutation. And again, in males, when they pass them on, whether large or small, they tend to stay around the same size. The last thing I'll mention here, because people have been hearing about AGG interruptions, many families who've been in our Fragile X community for a long time know about CGG, and then in recent years have been hearing about AGG. So the thing to know about AGG interruptions is that these are a small number of trinucleotides, again, 3 different chemicals that together may be found in that series of CGGs. So in other words, you may have 60 CGG repeats, and then maybe interspersed in there, you've got 2 AGGs. And so they're kind of interrupting that long chain of CGGs. And when that happens, and when you have these AGGs, which is a different chemical, adenine, guanine, guanine, it's a different trinucleotide, if that's in there, it acts as an anchor, and it tends to stabilize the gene and decreases the chance that it's going to expand any further up into the full mutation range. So that becomes important for women who have relatively small or mid-sized premutations. So let's say a woman who has a premutation of 70, and we would want to look at her AGGs, because that'll tell us if she has no AGGs at all, she may have a certain risk that that's going to expand to a full mutation. However, if she's got 3 AGGs stabilizing her gene, her risk for expansion may be much, much lower than another woman with the same CGG repeat size. So that type of testing is available, and really, though, as a genetic counselor, the situations in which we would discuss AGG testing is usually for a couple where the woman is considering having more children in the future, and she wants to really refine what really is the chance that this is going to expand. Because right now, the numbers that we give women are based on the overall population of women who have premutations and full mutations, for example. So on this chart, what you can see is that based on just across the board, not paying any attention to AGGs, women who have repeat sizes of premutations within the 55 to 59 range, they have about close to a 4% chance, or a 3.7% chance that that would expand to a full mutation in a pregnancy. On the other hand, if you're talking about somebody who's got a repeat size between 70 and 79, her chance is about 31%, so significantly higher that it would expand into the full mutation. Now keep in mind, in all of these repeat sizes, these premutation sizes, they're likely to expand, but they may not expand up until the full mutation range of over 200. As I mentioned, once you start getting over 100, you're close to 100% chance that that is going to expand into the full mutation range. So there's really not a whole lot of reason for women with these larger premutations to have AGG testing, because even having 2 or 3 AGGs is still not going to prevent that from expanding to the full mutation range. But for women down here in this range of, 55 to even up to like 89 premutation, size premutations, then they may want to have AGG testing, because if they have 0 AGGs, their chance of expansion may be much higher than somebody who has 3 AGGs interspersed in their chain of CGGs. The way I think of it sometimes, and it's a good parallel, is that the AGGs are kind of like fence posts. So imagine you had a big long fence, and you didn't have any fence posts along the way to stabilize it. It would just be some boards that wouldn't be able to, hold itself together as a fence. But by putting those fence posts in there, it holds it together and it stabilizes the fence. So we can think of the AGGs in that way. The numbers of AGGs are usually quite small. If they're present, they may be an indicator that the chance of expansion is much lower than if somebody doesn't have any. The last term I want to clarify is what's called mosaicism. So mosaicism implies that there are two or more different sizes of CGG repeats or methylation patterns in the same person. Now, I'll back this up by saying mosaicism also occurs in lots of other genetic disorders. And what this means is just that there are some cells that will have one genotype or a certain pattern, and others that'll have a different one. Now, typically in our genes, all of our genes and all of our cells have the same complement of genes. But sometimes, and particularly in Fragile X, what happens is as cells start to divide, further expansion might happen. So in other words, you might have somebody who has a Fragile X full mutation in some of the cells and Fragile X pre-mutations in other cells. And that can be detected. And even you may well have somebody who has a Fragile X full mutation, but they're of different sizes. The CGG repeat sizes are different in different cells. And so we call that mosaicism, when it's not all just the same in every single cell. The other important word you need to know when thinking about the full mutation in particular has to do with methylation patterns. Methylation, as I mentioned earlier, is a chemical tag that silences gene function. And when you have a Fragile X full mutation over 200 repeats, what happens is in that large expanded repeat series, you have the attraction of these methyl molecules, and it becomes methylated. So there's other chemicals that start to kind of attach themselves to this, and then the gene is not functional. So you can have mosaicism for size, as I mentioned, different types of sizes of CGG repeat patterns in cells, but you can also have methylation mosaicism. It's not as common to have methylation mosaicism, but what that means is in some cells the full mutation is methylated and can't produce its protein, which is what usually happens with Fragile X full mutations, but in other cells, for unknown reasons, there's a full mutation, but it's not fully methylated, so it's producing some protein. About 15 to 20% of males with Fragile X syndrome have mosaicism. Usually it's for size. They have different sizes of repeats in different cells. And then very occasionally you might have someone who's got methylation mosaicism. So they have different levels of methylation of their full mutation and may be producing some protein. So what we can say about mosaicism is, as a group, if you took 100 people with Fragile X mosaicism for full mutations, for example, they tend as a group to have higher functioning and to have fewer symptoms. So it's possible that they're producing more protein, either because they have repeat sizes that fall into the pre-mutation range and are producing some protein there, or they have a methylation mosaicism where some of their cells aren't methylated and are producing protein. However, this can't be predicted on an individual basis, and this is really important because you may have a very young child with some developmental delays who gets Fragile X testing done, and let's say it's a 2-year-old boy, and we find out that that's a boy who happens to have methylation mosaicism or a size mosaicism. Well, what I can say as a genetic counselor is we know as a group, generally speaking, they tend to have higher functioning and fewer symptoms, but on an individual basis, it's very possible that that young child will look very much like a child without mosaicism who has a full mutation. And it's because in the group there's a lot of variability, and even within the mosaic group there are people who are certainly functioning at the very low end of cognitive functioning, for example, and may have autism, and then you may, within that methylation group, that mosaicism group, have ones at the high end who are very high functioning. So it does tell us that maybe there's some residual protein going on, but we can't absolutely predict. We have no crystal ball to say how any individual child will do. So before we go to some questions, I'll just give you a few quick scenarios of how we diagnose Fragile X and what are the indications these days for Fragile X testing. Fragile X testing is very, very widely available. It's available through the major national commercial labs that are out there. It's very, very widely available, and the majority of health insurance plans will cover Fragile X testing for certain indications. So what are those indications? Well, for diagnostic testing of any child or adult, male or female, who has an undiagnosed intellectual disability, developmental delay, or autism, it is recommended that these individuals have testing for Fragile X. It's considered what we call a first-tier test. It is recommended by the American Academy of Pediatrics. It's recommended by the American College of Medical Genetics, National Society of Genetic Counselors. It's a relatively inexpensive test, and it should be first tier. There's no question. And unfortunately, we do know that sometimes kids wait a long time before they get diagnosed, and many people never get diagnosed. So it really needs a lot of attention to try to make sure that doctors are understanding what the recommendations are. Now, if somebody has a family history of Fragile X syndrome or even undiagnosed intellectual disability, developmental delay, or autism, carrier testing is available for those individuals, and carrier testing is highly accurate, as is the diagnostic test. There is prenatal diagnosis when a mother is a known carrier of Fragile X, and again, highly, highly accurate testing that can be done during pregnancy. I will point out here that there's a type of pregnancy test, genetic pregnancy test called NIPT, or non-invasive prenatal testing, sometimes called NIPS, non-invasive prenatal screening. That's done on a blood test on the mom during pregnancy, and that test, which is now widely offered to check for Down syndrome, for example, does not check for Fragile X. It will not pick up Fragile X. It will not even pick up increased chance for Fragile X. So in order to have prenatal diagnosis for Fragile X, it would take an invasive test such as an amniocentesis or chorionic villus sampling. Another group of people who are being offered Fragile X testing are women with infertility or unexplained early menopause. So a lot of the major infertility clinics, or fertility clinics, I should say, in reproductive medicine are offering Fragile X testing for women who present with infertility. And then we're now seeing diagnostic testing for individuals with late onset tremors and balance problems or ataxia of unknown origin. So for example, adult neurology is starting to test for FACS-TAS when they see, for example, a 57-year-old man who presents with some tremors and unexplained balance problems that were never there previously. It's not widely being done in neurology, but the word is starting to get out, and this is now considered a type of test when you have those kind of atypical symptoms, early onset Parkinsonism, etc. So let me show you what some of the ways we can see or diagnose families with Fragile X associated disorders. So any of you who have been to see a genetic counselor already knows that one of our skill sets is to draw out families trees using circles and squares. The circles are the women, the squares are men. These are the partners with a line between them, and these are the children. So this is what we call a pedigree or a family history. And in this family tree, let's say there's this little boy here, and he's coming for genetic testing or genetic counseling because he has a behavioral diagnosis of autism. Autism is a behavioral diagnosis for which there are many, many different causes. Fragile X is one of the most common causes we know. So we find out when I take the family tree that he has autism, but he also has a first cousin who has an intellectual disability, what we used to call mental retardation. And so that's mom's sister's boy. And then there's another aunt who has an anxiety disorder for which she receives treatment. And then if we go back in the history, what we find out is mom's mom, so this little boy's grandmom, has bipolar disorder. And then she had a sister who had infertility and didn't have any children. Now, I've colored these in different colors because really up until fairly recently we would think these were all unrelated conditions in this family, and yet when we do the math, so to speak, what we find out is that grandmom and her sister both have premutations which can explain some of their symptoms here. This little boy's mom has a Fragile X premutation, and it expanded to become a full mutation in him, so he has Fragile X syndrome. Mom's sister has a full mutation, so she had an expanded FMR1 gene that she got from her mom, and it expanded to a full mutation. She doesn't seem to have any symptoms that she's complaining about particularly, but she does have a son, and she passed her full mutation onto that son, and he has an intellectual disability. And then this sister over here also inherited a full mutation and has an anxiety disorder. So this is a kind of typical family tree that we might see in Fragile X. In this family tree, this is a grandpa who's maybe gone to a neurologist because he's starting to have some difficulty with walking. He's starting to notice some shakiness or tremors in his hands and balance problems. And he may get diagnosed with an atypical or unusual kind of Parkinson's disease in neurology. What we know is that he's got 3 daughters, and then he's got a grandson, and this grandson was just recently diagnosed with autism, and one of his daughters has an anxiety disorder. Now, through neurology, we find out that he has actually got a Fragile X or FMR1 premutation, and so he actually has FACS-TAS, and his symptoms are consistent with FACS-TAS. And then after he learns that, what we know, without even testing them, frankly, because we know that fathers always pass their Fragile X or FMR1 gene or the X chromosome to their daughters, all of these disorders, all of these daughters do have his premutation. And then we also know that this daughter with the premutation has a child with autism, and it turns out that when it got passed down this next generation, it expanded to a full mutation. So when I say that we don't have to test these daughters, we still do test these daughters just to confirm. Certainly there could be questions of non-paternity, something we deal with every day in our genetic counseling world. And not to say anything about this grandpa here, but he has 3 daughters, and really if we didn't test them, we could still infer that they are carriers of the Fragile X premutation. We can also infer that although it's very common to have anxiety for many different reasons, it's very likely that the anxiety disorder in his daughter is due to her Fragile X premutation. And here's a family tree, like I was telling you about before, where you don't have to have a family tree of anything going on really in order to have Fragile X. It can just be traveling in the family and not really showing a lot of problems for quite a while. So here we have a woman who presents to a fertility specialist because she's been trying to get pregnant for several years and has been unable to do so. And she finds out that her infertility is due to low ovarian reserve. She doesn't have that many eggs. And so when they do Fragile X testing, as they do at a lot of fertility clinics, it turns out that she's got a premutation. Well, when we go back in the family, it turns out that her sister also has a premutation, has a son who's developing typically, and then there's another sister who on testing is found to have a premutation. She is currently pregnant, and so the question and the concern is, what is going on with our child who I'm carrying? Did that child inherit a premutation? Maybe my other X chromosome that doesn't have the Fragile X expansion, or did it expand into a full mutation in that child? And then these 3 sisters, while all this is going on, are starting to think about their dad and remembering, you know, dad's really going through some personality changes lately. He's really not acting himself, and he's had some trouble. He's had a number of falls, and he gets diagnosed with FAXTAS. So Fragile X in families can present in many, many different ways. I want to acknowledge my colleagues at Geisinger in Central Pennsylvania at our Autism and Developmental Medicine Institute here, and in particular our Fragile X Clinic, which is part of the Fragile X Clinical and Research Consortium. So we'll take some questions now. We have we have some time left for questions, and I would like to just remind people that when you're asking questions, to keep in mind, because of HIPAA concerns, we certainly don't want to give away information about family members or name people or things like that, but any general questions that you have about inheritance, I'll be happy to take now.
Brenda, so a question came in, and I think I mentioned it to you earlier. Could you talk about the genetic testing 23andMe and why or why not they wouldn't, or should we approach them about including Fragile X testing? What are your thoughts on that?
Hi, Jane. So that question comes up. 23andMe is very well known. They do a lot of advertising, and 23andMe has been around for a number of years. It's a company that started off doing some direct-to-consumer or DTC genetic testing. And what makes 23andMe a little bit different than most other genetic testing companies is that they do not require a referral from a physician. So it's direct-to-consumer. And just a little bit of history, a number of years ago, 23andMe started offering carrier testing for a number of genetic disorders. There are thousands of genetic disorders. They were offering carrier testing for a number of disorders, including testing for certain breast cancer genes, etc. The FDA, the Food and Drug Administration, shut them down after a number of years because of concerns about offering genetic testing without any connection to a healthcare provider. And that was the concern, and there were concerns raised by the National Society of Genetic Counselors and other groups that, hey, you've got people out there, they're not getting any sort of context about genetic diseases or being told, where they can go for help or being given any information ahead of time to determine whether or not they want genetic testing, and so we don't think this is a good idea. The FDA agreed and shut them down. Recently, within the last year, so in the interim, I should mention, 23andMe for a number of years has really been doing ancestry testing, and so they've been able to use genetic testing to look for things that are not medical or health conditions. So, how much Neanderthal ancestry do you have in you, for example? And that is one of the things they look at. Within the last year and a half or so, the FDA has approved a limited number of genetic tests for 23andMe to do, carrier tests, etc. They also have increased the visibility of how you can get genetic counseling help through 23andMe, etc. So I give you that background, and I'll put it in a bigger context. There are many, many, many academic and commercial labs out there which have been offering Fragile X testing for decades at this point. It's a highly reliable test. It's not a difficult test to do. It's a relatively inexpensive test, and most health insurers in the United States pay for this. So, and this is my opinion here, this is not the opinion of the National Fragile X Foundation or of my employer Geisinger, but I will tell you that if I was going to have somebody looking to get Fragile X testing, I would have them go through one of these well-established genetic testing labs rather than a direct-to-consumer entity. The reason being, in order to do it through a typical lab, you do have to have some connection with a physician or healthcare provider or a genetic counselor. A referral has to be made. And I still do believe, and the National Society of Genetic Counselors continues to believe, that very often for certain types of complex genetic disorders, and Fragile X is one of them, that you really shouldn't be doing this in a vacuum and having people in the general population just sending spit in and getting a result without any connection to a healthcare provider.
Well, so speaking of spit, so how accurate is saliva testing?
That's a great question. So 23andMe isn't the only group that does saliva testing now. There are a number of different companies that are offering saliva testing for Fragile X. And this is really helpful, obviously, if you have a child or an adult who's really squeamish about blood tests and is not going to cooperate. The thing about the saliva testing, if they can extract the DNA in enough quantity to be able to analyze it, the test is as accurate as any other Fragile X testing. The problem with saliva is a lot of times there are contaminants in there so that they do not get an adequate sample to be able to test. And so the gold standard for any type of genetic testing is always blood. The result is more clean, if you will. It doesn't have other bacteria. It doesn't have other types of DNA, for example, from bacteria from something that might be in your mouth that could be unrelated, could be the piece of meat that you ate an hour ago, etc. So saliva, if they can extract the DNA and they can test it, it's absolutely reliable, but it is much more prone to having a failure and having to resubmit other samples and then ultimately going to blood. I will tell you, we definitely have used saliva in families where, let's say, there's an adult and he's extremely needle phobic. We already know Fragile X is in the family, and we're just looking specifically for Fragile X. If it's a child with a global developmental delay, and we're going to do a lot of genetic testing, not just Fragile X, but microarray and other things on that child, we're always going to try to get the blood before we'll send up a saliva sample.
So with those kind of contaminants, do you get false positive or false negatives, or?
It's not so much the false positive and false negatives. It's just, as they would say in the lab, noisy. It's not really clear. And so what you might get is, please submit another sample, and so there's back and forth with that. Now, it's gotten better. I mean, things have definitely gotten better. I'll give you another example of kind of ways around this is there's a large lab that instead of using just a swab inside the mouth, actually has people drink and they won't swallow it, but they'll drink scope, which kills off a lot of contaminants, and then they spit the scope into the test tube and send it up. And one of our problems with young kids with developmental issues, and even older ones, is a lot of times they don't spit very well, and so even trying to get that kind of sample is difficult. So I would just say you're not going to get a false positive or a false negative per se, as much as you're going to get just the sample wasn't able to be analyzed.
Got you. So the follow-up question to that is, some family members are overseas, and I don't know what this means. Having the FDA chain is useless. How can we approach the company to add Fragile X on the list? What is the regular process? So what I'm
So,
hearing you, yeah, go
yeah.
ahead.
Yeah, I'm not quite sure what they mean by the FDA chain is useless. I don't understand. The Fragile X is an approved drug, approved test. It's certainly approved as a medical test. It's highly, highly accurate. It's been around for decades. Trying to get it approved for 23andMe, specifically for that company, I think there's other efforts we should be doing. I'm not quite sure why people are a little fixated on 23andMe. As I say, that's a direct-to-consumer company. The test is widely, widely available through many, many other commercial labs and has been for decades.
Okay. So you can let me know if you have more questions on that, the person who sent that in. So here's another question. Do you recommend methylation analysis for all sizes on the premutation? Why or why not?
On the premutation?
That's what it says. Yeah, methylation analysis for all sizes of premutations.
No, I mean, and again, I'm a genetic counselor. I'm not a laboratory expert, but I will say that typically we don't worry so much about methylation when we're talking about premutations. Methylation comes into play when we're talking about full mutations because methylation is the process whereby the gene is shut down and not producing protein. So as a matter of course, if you find a premutation, typically you're not going to be carrying out methylation analyses and you're not going to have a result in that way. It's when you have a full mutation, and typically in Fragile X testing over the years, what you would find is that they would count the number of CGG repeats, and they would do a dual process using a laboratory technique called PCR, and then also one called Southern blot, and one would look at the number of repeats, and the other one would check the methylation status. So that's typically something that you would do in a Fragile X full mutation just to see whether the methylation was actually confirmed, meaning that the gene was not producing protein or was shut down by the methylation.
So anyone who gets a full mutation, the lab report should also state whether or not it's fully methylated?
Well, that varies from lab to lab somewhat. There are different types of Fragile X tests. So there is one that gets used very widely that's called a reflex to the methylation test. So they'll first check the number and then they'll look at the methylation status, or vice versa in some cases. But it's really a matter of doing both of those. But in recent years, I've noticed that there have been reports that really will just report out the number, and it'll just be said to be over 200. They won't even specify what the actual number is, and then there is no checking of the methylation status. So it's really going to depend on your geneticist, your genetic counselor, the lab they use, and what their preferences are. I think the bottom line is Fragile X testing is very, very well established, and it is not the type of test that we typically question when we get a result back. It's pretty accurate no matter what combination of methods they're using.
So, but if the only thing they're reporting is that it's over 200, then do you imply that it's also fully methylated?
Again, I think most geneticists and genetic counselors would want to see that the methylation study was done, but if that was only done and they counted the number and they didn't report that out, that is something I definitely would discuss with a geneticist. Chances are you can imply that it's fully methylated because most of the time it is fully methylated, but again, the other question about the methylation is in a full mutation, is it really good, even if you did find out that, there was some partial methylation for any individual child, that child's going to be your guide as to the severity of Fragile X syndrome, not so much the lab report. So again, I'm not going to second guess geneticists and other specialists who kind of make the determination of what tests they're going to use. The tests are highly accurate, and I think we're at a point now where, different labs are offering different combinations. But again, the gold standard for many years has been you want to know not only the number of repeats, but also the methylation status. Oh,
and then I think you and I had discussed this at one time. When someone is in the premutation zone, are there certain, is the AGG testing done automatically, or does that have to also be requested?
That is also has to be requested, although the technology that
Hello?
was, hello, hello?
Yes,
We can hear
I
you,
can hear you.
Brenda.
Okay, you can hear me. Yeah, so the company that developed the AGG assay and the ability to check those AGGs and make it clinically available has had that test available for a number of years. And the technology is also able to count the number of CGG repeats and look at the premutation. So some labs, it's my understanding that some labs are really moving toward that type of technology, which would at the same time check AGGs and CGGs, but most labs have not yet turned over to that type of technology. So I would definitely ask about it. There are certain instances where knowing your AGG is much more important than in other situations. For example, if you're talking about a boy with a full mutation, fully methylated full mutation, really knowing his AGG status is not particularly important. But if you have a woman with a CGG number around 70, let's say, and she wants to have more children, certainly she may want to know what's my AGG status. And then there are ways to refine her risk, whether she's at higher or lower risk than sort of that general risk we give. So I think the bottom line is to ask about it if it's not already presented to you. Some labs are combining them, and many labs are, it's a second test that needs to be requested.
Got you. Thank you. Okay, do we have time for a couple more questions?
Sure.
One of them is, one of my exes is 53, the other is 63. Does this mean it came from both my parents?
So that's an interesting question. You know, Fragile X premutations are pretty common in the population, but they're not that common. I mean, they're still relatively rare. I'd be a little suspicious about that result, and I'd want to just make sure, and if the parents were alive, I would check them. I mean, it's not out of the question to have somebody who maybe could have inherited a premutation from each parent. One of the things I would question is this woman actually got some mosaicism going on and has 63 and 53 that are both from the same parent, and then where is the second X chromosome? And so I'd want to double check into it. It's certainly theoretically possible, but it would be fairly uncommon.
I would agree. I think I've only spoken with one or two parents over the years who, or one or two moms who had, you know, both their exes were in the premutation range.
Mm-hmm.
Okay, it looks like one last question. What do you suggest for full mutation Fragile X girls who may be thinking about having children?
Well, that's a great question, and that could be its own webinar. Jane, you and I have done a series of presentations on sexuality over the years at the Fragile X conferences, so this is something that we do get asked quite a bit. Well, first of all, from a purely genetic standpoint, a woman who has a Fragile X full mutation has a 50-50 chance of having a child with a full mutation. There's no question of it, you know, is it going to expand or not like you would have with a premutation. If she's got a full mutation, it's going to be a full mutation in half of her eggs. So then half of her children, half of her boys, half of her girls who are conceived will have the Fragile X full mutation. We'd expect those boys to have Fragile X syndrome. We'd expect a large percentage of those girls to also have Fragile X syndrome, and the other to have maybe some other learning types of issues. So from a genetic standpoint, there's that chance of it happening. There's also the question, you know, if this person has Fragile X syndrome and has some developmental disabilities, is she going to be able to care for a child? And of course, those are things that can only be answered in knowing who the person is. And if you have a child with special needs, you really need to have that extra amount of parenting expertise. So whereas a typical child may not be as much as a challenge for a woman who's got intellectual disabilities due to Fragile X, would she be able to handle a child with extraordinary developmental needs? So those questions have to be there. And so, you know, I think in respect to adults, whether they're male or female, who are making reproductive decisions related to Fragile X, they really should meet with a genetic counselor or someone who can speak with them about these issues. I can give you an example. A young woman who I've known since she was 12, and she's now in her 30s, and we had talked even starting as a teenager when she was going to school at my former employer, where she was receiving special education services. We would talk about her interest in getting married, her interest in having children, etc. She ultimately did get married, and as a young woman, knew that she had uncles with Fragile X and a brother with Fragile X syndrome, and she had a lot of difficulties herself, wasn't able to drive. She was able to hold down a job, but she also was having some economic difficulties with her husband, and she just decided, the chance that I would have a child with Fragile X would really be a challenge for me. And she made the decision after lots of counseling that she did not feel that having a child was that important to her. There are other women who may decide otherwise, who may decide that this is what they really want to do. They want to be a mom, but they need to have those conversations to determine what kind of supports need to be in place to make this successful. The last word on that I'll mention, because I talk to a lot of family groups, and one of the things, if you have very young children and you're thinking, I'm really concerned when they get older, I don't believe that they're going to have the capacity to independently parent a child. I mean, I think the message should be, and maybe this is true for all children, typical as well, that, it's not the be-all and end-all to have kids. In our society, in a way, if you don't have kids, it's all, in a way, you may feel like you failed. You haven't done what you need to do as an adult. But I would say from a very young age, for children with special needs, to give them other alternatives and point out people, look at this person, she's a single woman, she did this, or look at this married couple, they didn't have children, but look what they do, they travel around, they have 7 cats, who knows what we want to say about them. But I say this to my own typical children because I don't want there to be any expectation that somehow not having a child is lesser. And I do think that that's an important message that we should all be bringing home to our kids.
I really like that. Now, one more question's come in. It's and if you have time, we'll ask it. Otherwise, I can pass off. You want to take one more?
I have plenty of time, sure.
Okay. My son, who's 41, has a full mutation. Because of my belief that my father's intention tremor was sure to be fax-tas, I think that's what my father received a blood test. My father was highly intelligent, a leader in the community. It turns out my father has a full mutation in the 200s, unmethylated. How did this happen, and does this mean I have the same type?
Well, that's a great question, and I think for the person asking this, then, it sounds like her father was tested, and the answer to that would be for her to have a test done as well to see if she has the same type as her father. So, the cutoff of 200 is not a biological cutoff. It is a cutoff that's been observed that above that point, we have what we call a full mutation. But again, biology isn't calling it a full mutation and a pre-mutation. So it's possible that there are individuals who hover around that 200 mark who either maybe even below at 195 repeats, for example, might have a fully methylated gene and might have Fragile X syndrome. And there may be those above 220 around that area who actually are more like a high premutation, and they don't get methylated. And she said he's unmethylated. If that's the case, we would expect normal or close to normal protein production, and we would not expect Fragile X syndrome. So it sounds with her father that it's possible that he may have a high premutation that happens to be in the very low range of full mutation, and he has fax-tas. The other possibility, and again, I mentioned mosaicism before. Mosaicism should be looked at in that situation because it's possible that he has some mosaicism for true premutations under 200, and he just happens to have some over, and maybe functioning fine. I mean, genetic testing, those would be cases, because they are unusual, where I think I would engage the support of a geneticist or a genetic counselor to look very, very closely at the genetic testing. Genetic testing is highly reliable for Fragile X, but when you start getting into those unusual situations situations, somebody with a full mutation who's unmethylated, who's a small full mutation, I would want to know, should we rerun this test? Should we be looking at it? What things should we be thinking of? So in other words, it does sound like, in that family, the real answer is that he's been tested and he's unmethylated, and that's why he's functioning as high as he is.
Okay. I think, one more question. Do repeats ever decrease through generations?
They can. We call those contractions, and contractions can happen fairly regularly, especially when a father has a premutation and then he has daughters, and then we find that those daughters have somewhat smaller repeat sizes than their dad. We also see this when it happens sometimes from moms to their sons or daughters. Usually, though, contractions are small. It's a few repeats and typically under 10 repeats. You don't usually see very large contractions of dozens of repeats, and certainly we haven't documented anybody with a large contraction going from a full mutation in a mother, let's say, down to a premutation in a child. So it doesn't usually go backwards that way, but there can be small, usually under 10 CGG repeat contractions that happen from one generation to the next.
Okay, I have seen that. I guess, and this prompted another question for me. What's the smallest repeat that you're aware of that went to a full mutation?
Yeah, so that the smallest published repeat that went to a full mutation is, I believe it was 59, if I'm not mistaken, but we have the, and part of that is how we set up these categories, right? So 59 there was a published case of somebody with a 59 whose repeat went up into the full mutation range. We set the cutoff for premutations at 55 because below that we really have never seen such very large expansions at all. So that's where you get that gray zone or those intermediates. I believe it's 59. I could have missed some papers that actually were a little bit lower, but certainly not below 55.
Okay. Well, Brenda, I think with that we will we'll call it a day. I would like to thank you for sharing your expertise with us. I mean, you have a wealth of information about genetics. I learn something every time I hear you speak, and I'm so thankful for that. As a
Thanks, Jane. It does sound like people do have questions, and again, I'm serious when I say, I talk with families who've had the diagnosis for years, and they're feeling a little sheepish because they don't really understand the genetics, and I just want to empower people to say, hey, this is something genetic counselors struggle with. It's a very complex pattern, so speak up and ask your questions. No question is stupid at all.
Well, and I think that, as you learn, as you increase your knowledge level, then you wonder about something else, and then you wonder about something else, and so it's always coming up. So I thank you. I mean, this is we have you on about once a year, and I'd love to continue to do that because there's always questions and there's always new information.
Very true.
I want to let all the registrants to the webinar know that you will receive a recording of this in an email in the next week or so, and it will also be on YouTube within the next week. Before I sign off, I want to let everyone know these webinars are made possible because of your contributions to the Foundation. Please consider making a contribution at fragilex.org. The next webinar will be in July, early July. Right now, it's scheduled for July 6th. And it's Strategies for Girls with Fragile X Syndrome, and it will be presented by Barbara Haas-Givler, who works with Brenda, matter of fact. So watch your emails for further information on registration. And as always, please let me know if you have any questions. I'm Jane at fragilex.org. And Brenda, do you have any parting comments for the group?
Well, no, I don't know how many are out there in the group, but thanks for listening in, and please reach out to Jane and to the Foundation if you have additional questions that you didn't get answered today.
Great. And there you have it, today, May 16th, 2017. Thanks, everyone. Have a great rest of your day. Thank you, Brenda.
Thank you.